Welcome to the biology class. In the previous video, we discussed about gymnosperms. In this video, we will learn about angiosperms. Gymnosperms and angiosperms come under the group spermatophyte. Spermos means seed, phyte means plants. Gymnosperms and angiosperms are seed-producing plants. But in angiosperms, the ovules are enclosed inside the ovary. In gymnosperms, ovary is absent, so ovules are naked. Angiosperms are commonly available plants. What we see around us are mainly angiosperms. They are adapted to the terrestrial mode of life. Angiosperms have well-developed vascular tissues, so they are adapted to live on land. They constitute major group of our Earth. Angiosperms evolved 140 million years ago. In angiosperms, sporophyte is the dominant phase. What we see as neem tree, coconut tree, banyan tree are sporophytes. The sporophyte is a dominant phase. The sporophyte produces spores. Pollen grain is microspore. The sporophyte lives for long duration and gametophyte is very simple. It lives for short duration. Vascular tissues, xylem and phloem are well developed in angiosperms. Flowers are produced instead of cones. In the previous group of plants, cones are produced. The reproductive structures are found on the cones. But in angiosperms, the flowers have male and female reproductive structures. The ovules remain enclosed in the ovary. Ovules are protected inside the ovary. Pollen tube helps in fertilization. When pollen grain falls on the stigma, the pollen tube carries male gametes through the style and reaches the ovary. Double fertilization takes place. The pollen tube carries two male gametes. One male gamete fertilizes the egg and forms a zygote. The second male gamete fertilizes a diploid nucleus. Male gamete is haploid. The second male gamete fertilizes the diploid nucleus, so a triploid structure is formed. The triploid structure has three sets of chromosomes. That triploid structure divides and forms the nutritive tissue called as endosperm. Endosperm develops from a triploid cell, so it is also triploid. Angiosperms have two groups. They are dicotyledons and monocotyledons. Dicotyledons have two cotyledons in the seed. Seeds of dicot plants have two cotyledons. They have reticulate venation. The veins branch and form a network in dicotyledons. Dicotyledons have taproot system. The radicle forms a main root. The main root has branches all together form a system called as taproot system. They get tricolpate pollen grain. That is, the pollen grain has three furrows. Flowers are tetramerous or pentamerous. This flower is a pentamerous flower. It has 5 petals. Pentamerous flowers have floral pods 5 in number. They have 5 number of floral pods in each whorl. 5 or multiples of 5 members in each whorl of the flower. That is pentamerous. Tetramerous flowers have 4 floral members in each whorl or multiples of 4. Monocotyledons have single cotyledon in the seed. They have parallel venation. Parallel venation is very clearly seen in grasses and banana. Monocotyledons have fibrous root system. The radicle is a short-lived structure. In dicots, radicle forms a primary root, main root. In monocots, the radicle is short-lived. Cluster of roots arise from the base of the stem and form a fibrous root. Radicle doesn't persist, it is short-lived. Monocolpate pollen is present. The pollen has single furrow. Flowers are trimerous. The floral parts are 3 or multiples of 3 in trimerous flowers. Let us see the differences between monocots and dicots in their internal structure. Vascular bundles are arranged in the form of ring in the stem of dicotyledons. In dicotyledon stem, vascular bundles are arranged in the form of ring around the pith. In monocotyledons, vascular bundles are scattered in the stem. Vascular bundles are open. In open vascular bundle, cambium is present between xylem and phloem. The cambium is a meristematic tissue. It provides new cells. In monocotyledons, vascular bundles are closed. There is no cambium in between phloem and xylem. Because of the cambium, secondary growth is present in dicotyledons. The cambium produces new cells, so increase in the thickness of stem and root takes place. Increase in the thickness is called a secondary growth. Monocotyledons have no cambium, so they do not show secondary growth.